That is the key difference which drives us to think if can we leverage these endogenous pathways to develop the recyclable lactate. So basically we connect the two disposing systems together. One is the micropinocytosis and the second is the lysosome-mediated endocytosis. We don't want to exclude every possibility of this technology. So we are also trying to see if we can start up a company to translate this technology, but we are also willing to work with other established companies who is very interested in our technology as well. Human disease are always associated with dysregulated proteins. Protein degradation is trying to use small molecules or antibody-based degradation tools to basically just grab the proteins and by hijacking some endogenous protein disposing system in the cell to degrade the proteins. For example, for the lactate technology, which is lysosome targeting chimeria. This is a groundbreaking technology that have a huge potential in treating different disease. So it has been applied into different disease treatments such as cancer and the autoimmune disease. But the major weakness of this traditional lactate technology is that for the traditional lactate, it will degrade together with the targeted protein, actually impeding the clinical translation of the lactate technology. And our innovation is trying to address these challenges, try to expand the clinical application of traditional lactate technology.
So our invention is to design a protein degrader which can be recycled by the cells to maintain a sustained protein degradation. So we just use a very simply method by the cells that's called the micropinocytosis. So this is a kind of natural or endogenous cellular transport pathway. So during this process, the substrates cannot be degraded or changed. So we think we can use this transcellular pathway to do something, to recycle our protein degrader. So when we came up with this idea, so we are very excited because we think it's a very simple and straightforward strategy. So it's very promising.
Our new innovation, when the lactate technology bind to the proteins and they go into the lysosome pathways in the lysosome or in the pre-lysosome, we call them micropinosomes. So these lactate tools will disassemble with the targeted protein, or they will separate from the targeted proteins, and then the lactate technology will be recycled back to the outside the cells.
We are excited to find out that we get a sustained protein degradation in vitro, and this really encourages us to further validate its efficacy in vivo. So we do the mouse model of the breast cancer. So we find it can really achieve long-term protein degradation by just 2 injections. So this is our inventions and how it works.
Since in this invention we already devised these new tools, now we are trying to make it much better and much efficient for the future clinical translation. We are trying to collaborate with the physicians in the Carbon Cancer Center, try to see how they view our technology and is there any potential applications that can be used in their clinical disease treatment. We like to work with 2 types of people. First is the startup company who is also working in the similar area but facing the challenges for the traditional lactate, and we feel like our inventions can help them to address this recyclability of the lactate technology. And the second, we are trying to working with more like clinicians and the physicians who see the potential of this lysosome targeting chimeria in disease treatment, which is a groundbreaking technology that's revolutionized the traditional pharmaceutical industry.
It's actually the first project since I joined this lab, and it can give me a lot of inspirations. So I'm now working on how to expand its application in different disease treatment, and also I want to find out its potential for many treatment strategies such as a tumor vaccine.
Hi, my name is Rafael Diaz, licensing manager at WRF. If you like what you just saw and want to learn more about these high potential technologies, please get in touch with us with information on your screen. And don't forget to like us and subscribe for more videos from WRF.